Everybody's always asking me about these shirts. So as we study animals now, I had the excuse to go out and buy another one, this one with little fish on it. Most primary teachers feel quite comfortable teaching a unit on animals. There are some major topics that should be covered. First, the diversity of animals. The fact that there's such a wide variety of animals in our world. By studying this diversity, allowing the students to have hands-on experiences with animals, they should develop a respect and appreciation for animals. And once again, you are the role model. How you treat animals will probably be how they will treat animals. If you're studying earthworms, for example, like this one I have right here, and after you're through studying it, you just go and throw it in the trash and say, it's just an earthworm, it doesn't matter. Then your students will start to feel that way about earthworms too. If, however, on the other hand, you go to great pains to find a nice plot of soft soil to give the earthworm at least a reasonable chance, then you will start developing this respect for animals amongst your students. That's very important. Some of the other topics you should cover are the needs of animals and the characteristics of animals, how one differs from another. Some of the characteristics to investigate are the size of the animal, are they big or small? What is their outer covering? Do they have fur, feathers, or scales? How do they move? Do they run, hop, swim, fly, or squirm? Where do they live? Perhaps in a tree, in the water, or underground. And finally, what do they eat? Are they plant eaters, animal eaters, or do they eat both? By learning about the characteristics of different animals, particularly the vertebrates, students should be able to begin to classify them into separate groups. There are 5 major groups that they should be familiar with: the mammals, the birds, the fish, the reptiles, and the amphibians. Let's take a look at some of the characteristics of each one of these types. Mammals have hair or fur. They breathe air with lungs, and they're warm-blooded. Birds have feathers, wings, and beaks. They breathe air with lungs and are warm-blooded also. Fish have scales and fins and tails. They breathe air with gills and are cold-blooded. Reptiles have horny scales. They breathe air with lungs and they're also cold-blooded. There are 3 fundamental body types of reptiles. There's the crocodile and lizard body type, the snakes, and the turtles. Amphibians live on land and water. They consist of frogs, toads, newts, and salamanders. They may either have a soft, moist skin with no scales, or it may be a dry skin, such as a toad has. They can absorb oxygen either through their skin when they're underwater, or with lungs when they're on the land. And they are also cold-blooded. An additional difference between the mammals and the other 4 types is that the young develop within the mother's body in the mammal and are born alive. And in the other 4 types, the young usually develop within eggs that are laid by the mother. You should be able to find examples of each one of these 5 types of animals and bring them in for close inspection by your students so that they can really realize the difference between each type. And when you're studying the characteristics of animals, you can start small. First classify animal pictures according to big and small, and then get more involved like fur, feathers, or scales, and maybe warm-blooded and cold-blooded. And by building as you classify, you should be able to almost trick the students into discovering these 5 main types. Now let's go on and do our first investigation, not in a vertebrate animal, one with a backbone, but one without a backbone, the earthworm. This will be an experiment in discovery. You can get these night crawlers from your local fishing tackle supply, and your students will remember this for the rest of their lives if you have a live earthworm activity. They may be a little squeamish about holding it, but it's certainly a harmless little guy, and as long as you don't squish it, you can't hurt it. You can take it out of the nightcrawler box and put it on a piece of paper towel. Each student can have one, or you can share one earthworm for 2 or 3 students. Make sure they have their magnifiers because they're going to just do simple observations. What can they learn about this earthworm? Where's the head? Where's the tail? Is it segmented? Can you see the little vein running down the top of the body? Is the bottom of the earthworm the same color as the top? Of course, as you see here, the bottom is much lighter in color than the top is. This other piece, this center piece, is called a cotellum, and it's located closer to the head end than it is to the tail end. This is actually a reproductive organ of the worm, and when they reproduce, they have both male and female sex organs inside each worm, so they line up end to end, head to tail, and cross-fertilize each other. If you have an advanced class, you can have them try and count the number of segments from the cotellum to the head end and see how many people get the same number. Remember to keep the worms moist. As they dry out, brush them with water every now and then. And as they get kind of lethargic and worn out, it's probably good to put them back in the container and store them in the refrigerator when you're not using them. And finally, remember that when you're through using your worms, if they've survived all the screaming little kids, then you can put them back in the soil and have them actually help some plants out. Another fun thing to do is to have your worm activity on the day they're serving spaghetti. There are other activities that your students can do to learn more about the earthworm. One is to see how they react to light. To do this, get the long test tubes. These are the 200 millimeter variety, or you can get an olive jar. Find a cork or stopper that fits in one end. Place the earthworm inside the test tube and cover half of it with aluminum foil. Expose it to light and see whether the earthworm likes the light part or the dark part. A good simple little experiment. You may also wish to show them what an earthworm looks like inside without actually harming the earthworm. To do this, first you want to demonstrate what you're talking about when you're going to be looking at it so that they can relate three-dimensionally to the length and the cross-section of the earthworm. Get a hot dog and cut it into a thin slice, cross-sectional slice, that looks like a wafer. Tell them that we're looking at the worm in that same type of cross-sectional view. And then go out and buy from a science supply house a microscope slide that has the cross-section of an earthworm. This is what the cross-section of an earthworm looks like, and it's so big it almost doesn't fit in the field of view even when we're at the lowest power. Let's take a close look at some of these individual body parts. First, we notice two different types of muscles, the circular muscles and the longitudinal muscles. When the circular muscles contract, they affect the worm's diameter. When the longitudinal muscles contract, they affect the worm's length. We also notice a main blood vessel. This is the blood vessel that we were able to see on our live worms. It runs the length of the worm's body. Also running along much of the worm's length is a digestive tube or intestine. This allows the worm to get nutrients from the soil as it eats the soil at one end and expels waste material at the other. Traditionally, the study of slimy little critters, like earthworms, has been left up to little boys. But you should make it your goal to create a universal interest in these lower forms of animals. Don't let those girls get off by going, "Eek, I can't touch that." Get everyone involved with these. Let's take a look at some of these other lower life forms, the invertebrates, that we can investigate. First, I have mealworms. They're very readily available from a bait and tackle store where you bought your night crawlers, or you can get them from a science supply house. When your students are observing their mealworm, you should have them put the magnifier very close to their eye and then move the sample in until it comes into focus. They'll be able to see much more than if they're holding it from a distance. Plus, it gets them in a closer proximity to the little animal and dispels some of that fear they might have about it. Some other good samples are snails, and you can see in the snails I've put numbers on the back of the shells with whiteout so we can always identify them and make comparisons. Sow bugs are also fun to watch. They're also known as roly-polies or pill bugs, and they don't like light. In fact, you can find them under boards in your backyard. If you put a piece of black paper over a section of it, after a few minutes they'll all tend to gravitate toward the dark area under the black paper. Crickets are fun to keep too, and you can find them at bait and tackle stores or at pet stores. When you prepare the samples for your students, you can put individual crickets in a plastic bag, seal it almost all the way up, insert a straw and blow it up with air, and then seal it the rest of the way. The students can observe the cricket inside their bag. It should be able to stay alive with no problem for about an hour. When you're looking closely at the crickets, you can tell the difference between the male and the female ones by looking at the tail end. The female cricket has a long tube. This is called an ovipositor and is used for laying eggs. Remember that all these little animals are not harmful at all. They can't hurt you. They just maybe get your hands a little slimy, and you can always wash your hands. As the students in your class investigate animals, remember that the activities you choose should not only challenge their process skills, but also concentrate on those areas I discussed earlier. The ones where we talk about diversity of animals, characteristics of animals, their needs, their habitats, and so on. This next one, where we investigate sea monkeys or brine shrimp, is very exciting and covers a lot of these areas. Here's what we're going to do. We're going to hatch brine shrimp. Sometimes they're called sea monkeys, and you can pick them up in egg form at your pet store. If you can't find them at the pet store, of course they're always available at a science supply house. In this experiment, we're going to investigate how the concentration of salt water affects the hatching rate of these little brine shrimp. Each student group should have access to some measuring equipment, some distilled water, and some salt. Have them measure out 100 milliliters of water, preferably with a graduated cylinder, and pour it into a clear plastic cup. If you don't have the graduated cylinders, just get the 10-ounce cups and fill them about a third full. Then each group will have assigned to them a certain amount of salt to put in their cup. Have one group put no salt in their cup, another group put 1 gram in, a third group put 2 grams in, and so on. Go up to about 10 grams per 100 milliliters of water. The best way to measure out the salt is to first weigh a piece of filter paper and then add the number of grams that you need of salt to that weight. This piece of filter paper, for example, weighs 1 gram. If I want to put 4 grams in this cup, I'll boost it up to 5 grams, so I'll have 1 gram, which is the weight of the filter paper, and then 4 grams of salt that'll be on top of it, which will total 5 grams. This is also good for working on the addition tables and some simple logic here too. So I'm going to measure out about 4 grams of salt on this, bring it up to the point where it's balancing, bend this a little bit, and drop it into this cup. Shake it in, stir it up with a spoon, and so on. Make sure you mark each cup with the amount of salt that's in it. This one here, for example, has 8 grams of salt. The next step is to add the brine shrimp eggs, and the best way to do this and be somewhat scientific is to allow the captain of each group to come up with a Q-tip in their sample and scoop out as many brine shrimp eggs as they can with the Q-tip. You can only hold about so many, and shake it into their cup. Once the brine shrimp eggs are in there, it takes about 1 to 2 days for them to start hatching. If you place these all together in the room where it's somewhat warm, 68 to 70 degrees, you'll have some of the best conditions. It might also be advisable to cover them with plastic wrap. Once they've started to hatch, we want to start making comparisons. Which concentration of salt water produces the best results for hatching brine shrimp eggs? Incidentally, the 3 gram solution is most similar to that of seawater. Allow your students to make a hypothesis as soon as they set this experiment up. Have them guess which solution they think will have the most productive results. After 2 days, allow them to go back to their samples and take a look. There's a number of ways that you can observe the samples. One of the most exciting ways is to darken the room and shine a flashlight through the top and allow students to walk by and look at the different samples. In my experiment, I found that none of the eggs hatched in the solution with no salt, nor did any of the eggs hatch in the solution that was very concentrated with 10 grams of salt. Only a few of the little brine shrimp were swimming around in the 8 gram solution, and in the 4 gram solution it was just teeming with life. The effect can be just as dramatic using the magnifiers by holding the sample up, putting the magnifier near your eye, and looking at all the hundreds of tiny little sea monkeys swimming around. Have your students make a drawing of what they think they look like, and then you take a sample with an eyedropper, put it on a well slide, and a well slide is a slide that has a little indentation in it. And I hope you all have access to a micro projector, because then you can show your students what these little sea monkeys really look like. You may even be fortunate to see one hatching out of an egg. As your students are studying these tiny little animals, be sure and have them demonstrate how the sea monkeys flap their little sea monkey arms and push or paddle themselves through the water. And while we have the microprojector out and we're talking about how little animals move, there's another very good animal that you can put in your well slide and take a look at. It's called the vinegar eel, and it's a very resilient animal. Lives for a long time inside this little container. In fact, my sample here has been alive for about 6 months. Let's see what they look like. These animals are actually not eels, but worms, and it's interesting to watch them squirm through the vinegar. You might discuss pasteurization, why vinegar is boiled before it's sold in the grocery store. And don't forget the wide world of prepared slides that are available from equipment suppliers. Why not look at a mosquito, or the leg of a housefly, or perhaps even the stinger of a honeybee? Prepared slides are inexpensive, or you can always make your own. The method for doing this is described in the 4-6 Life Science tape. Remember to involve your students as much as possible in the process of looking at these tiny animals. Have them put the drop on the well slide. Have another student focus the microprojector, and get as many students involved as possible in describing what the animal looks like. Now let's go to a little bit bigger animals and talk about classroom pets. It's both interesting and fun to have a pet in your classroom. It's valuable too as you involve your students with animal care. You might prefer low-maintenance pets like birds or fish. Incidentally, fish tanks make great homes for other pets besides fish. You can even fashion an escape-proof cover out of hardware cloth. The anole is a reptile that's easy to keep and fun to watch. It lives off mealworms or crickets and can change the color of its skin. Snakes are reptiles for the more adventurous teacher. These harmless garter snakes live off guppies that are swimming in the water bowl. Turtles also eat feeder fish. Their tank needs cleaning once a week, and they require a special vitamin D lamp. You may prefer a small mammal. Guinea pigs are always a classroom favorite. Rabbits require more attention, but wire cages are inexpensive and easy to clean. This guy is Rabbi Raboon, who spent some time at school and now lives in my backyard. Hamsters have a tendency to sleep all day. I've heard they can even eat and sleep at the same time. Regardless if it's mice or hermit crabs, there's a special animal for every classroom and a special treat in store for every student.